Jessica Howes is the Deputy Director of the Division of Systems Biology at the National Center for Toxicological Research at the FDA in Arkansas. The title of her talk today is The Role of Modeling and Simulation in Regulatory Decision-Making.
So first of all, thank you for the invitation. As you mentioned, I'm currently the Deputy Director in the Division of Systems Biology at the FDA's National Center for Toxicological Research. I've been here for about 1.5 years, but I do come with 10 years of review experience in the Center for Drug Evaluation and Research. Today, I will be presenting on the role of modeling and simulation, M&S, in preclinical and non-clinical regulatory decision-making. As a disclaimer, the content of this presentation reflects my own opinions and does not necessarily represent the official position of the Center for Drug Evaluation and Research, the NCTR, the agency, or the federal government. In this presentation, I will be talking about regulatory perspectives on the use of modeling and simulation in determining clinical doses for early phase trials, the influence of M&S on non-clinical bioassay study designs for drug products, how dose exposure response curves can influence dosing regimen decisions in early phase clinical trials, the role of M&S in drug product reformulations, and if we have time, considerations for drug products with significant accumulation potential. So so that we're all on the same base or on the same page, some of this may be review for many of you, but preclinical data is used to predict therapeutic windows for drug products where the difference between dose exposure response curves for therapeutic pharmacology, represented here in blue, and adverse toxicity or pharmacology, represented in red, is the therapeutic index. The MABEL is the minimally anticipated biological effect level for a given drug product and is usually predicted based on preclinical data, including in vitro pharmacology data from target cells and from human and toxicology species, from concentration effect data from in vitro and in vivo studies, PK and PK/PD and PBPK modeling. The ATD, anticipated therapeutic dose, is the predicted range of doses that are likely to result in 50% to 100% of pharmacological activity. The PAD, pharmacologically active dose, is the predicted dose for 50% pharmacodynamic activity. The low AEL is the lowest dose at which adverse effects are observed, and the orange shaded region here indicates evaluated doses with adverse toxicity. The no AEL is the highest dose at which there are no significant increases in adverse effects in comparison to the control group. The no AEL is determined empirically and must be based on GLP-compliant non-clinical bioassays to support clinical studies. Notably, M&S, modeling and simulation, plays an important role in predicting clinical MABEL, PAD, and ATD. Although toxicology endpoints include no AELs, low AELs, and MTD, which is the highest dose that does not produce unacceptable toxicity or maximum tolerated dose, the no AEL is the gold standard for first-in-human studies and can be a basis for the maximum recommended human dose. Pharmacology endpoints most often used for determining clinical doses include the MABEL and PAD. Since toxicology endpoints are actually the standard expectation, it is important to know when it is appropriate to use pharmacological endpoints. The most common reasons include when there are no relevant non-clinical species to assess activity of the drug product, when there are significant differences in PK/PD and biology between animals and humans, and when toxicities in animals resulting from exaggerated pharmacological effect can be associated with the absence of a no AEL. The FDA 2005 guidance for estimating first-in-human doses states that the PAD in these cases may be a more sensitive indicator of potential toxicity than the no AEL and might therefore warrant lowering the MRHD. The importance of these recommendations came to light with the 2006 De Janeiro incident in the UK, where all 6 subjects in the first-in-human trial for the monoclonal antibody TGN-1412, experienced cytokine storm resulting in serious and life-threatening adverse events, including irreversible brain damage and limb amputation. Thus, for some drug products, particularly biologics, one should consider the appropriateness of a pharmacology endpoint. When translating non-clinical doses at toxicological or pharmacological endpoints in order to determine human risk and safe clinical doses, it is imperative to apply a safety factor to compensate for species differences and unknown variables that may impact safety. The standard safety factor is usually a value of 10, which has been validated experimentally and is based on several observations, including the likelihood that humans may be more sensitive to the drug's pharmacodynamic activity, that some toxicities are difficult to assess in animals, such as headaches, myalgia, and mental disturbances, that the drug may have interspecies differences in protein binding, absorption, distribution, metabolic clearance, and excretion, all of which can affect overall bioavailability. There may also be differences in the drug target's densities or affinities, or even human-specific toxicities that simply aren't identified in the preclinical species. However, there are times when modifications to the standard safety factor of 10 may be justified, which could include either an increase or decrease. In fact, there are numerous potential scenarios where modifications can be justified. Examples justifying an increase in the safety factor above 10 include non-monitorable toxicities, irreversible toxicity, and unexplained death observed in the non-clinical bioassays. Modeling and simulation evaluations related to the steepness of dose-response curves and, I've got the animations backwards, nonlinear PK can play a role in determining if these justifications are applicable. Examples justifying a decrease in the safety factor below 10 include new drug products within a well-characterized drug class with similar PK, ADME, and toxicity profiles across species, including humans, which M&S can play a significant role in demonstrating. Also included are non-clinical toxicities unlikely to be translatable to or relevant in humans, and toxicities that are due to exaggerated pharmacodynamic effects in healthy animals that are less of a concern in the indicated population. Demonstrating that the dose response for toxicity is not steep. Sorry, just a moment, I lost my notes. Demonstrating that the dose response for toxicity is not steep could also support justification to reduce the safety factor, which of course M&S could play an important role in demonstrating. The 2005 FDA guidance for estimating the MRHD in first-in-human studies depicts this useful flowchart for determining the MRHD maximum recommended starting dose. However, it is important to note that this example is for systemic administration only, but there are additional guidances for other types of administration. As previously mentioned, the NOAEL toxicological endpoint is the gold standard for determining an MRHD. However, if the MABEL or PAD is deemed an appropriate basis for the MRHD determination, then that can and should be considered. The EMA 2017 guidance contains more information on this, which could be very useful. Once the toxicological or pharmacological endpoint for the most appropriate species is identified, it can be converted to the human equivalent dose, HED, using an allometric conversion factor to normalize for interspecies differences in body surface area.
Jessica,
However,
we're missing our slides. We're missing your slides.
Oh dear. I have no idea at what point you didn't see the slides anymore. I apologize.
It's been probably about 3 or 4 minutes now.
Oh dear. Let me know if you can see them again. I think my connection.
I see MRHD flowchart.
Yep, that's where I am. And I apologize for you not seeing the slides. Did you guys see the slide?
Yeah, we've seen
Okay.
this, and it's
Okay, good.
not.
All right.
If you can make it full screen or something, it would be good. Thank you.
Yeah, and you guys can get these slides afterwards too.
Okay.
There. Is that a little bit better?
Yes.
Yeah.
Okay. Okay, so moving on with the MRHD flowchart, I'll pick back up where I left off. I hope that's okay. So let
It
me
looks like
catch
if
up
you can
with
make it full screen, I don't think you can. Your PowerPoint does not seem it's full screen.
Is that better? Sorry.
It's better, but
Yeah, I need to have my notes and my other monitors are not connecting this morning. I'm having a few technical issues, as you can see.
maybe
The
if I
joys
could,
of working from home.
okay. Well, all right. I don't want to take your time further. Thank you, so.
Okay. So let's see, where were we? So in determining the human equivalent doses, it is important to note that if the drug is administered directly into the blood circulation via intravenous injection or bolus, then there is no need for the allometric conversion factor. The MRHD is then calculated by dividing the HED by the most appropriate safety factor and multiplying by the average human weight for an adult, which is still considered to be 60 kilograms by most regulators. Determination of the maximum recommended human dose, MRHD, for drug products is based on empirical GLP toxicology and safety pharmacology data as discussed in ICH M3R2 and is not based on PK modeling. It is appropriate to take into account target saturation of the drug target, where the MRHD should be within the estimated human pharmacodynamic dose range, in which MNS does play an important role, and should not include internal concentrations above which there would be an expectation for complete. Okay, great. So the point that I wanted to make here is that the clinical MRHD can be limited by the highest dose tested in non-clinical studies. And since MNS can play a role in how some sponsors and investigators choose the high dose that they want to investigate in those non-clinical studies, this is very important to keep in mind, because if that high or top dose, non-clinical dose, ends up being too low, it can end up crippling the clinical studies. The MRHD can also be limited by an insufficient margin of safety or exposure margin at the non-clinical no AEL and or low AEL for significant adverse findings. The basis for determining an acceptable MRHD in the presence of significant adverse concerns usually takes into consideration the severity and reversibility of the findings, as well as the indication, patient population, and severity of disease or disorder being treated, and is determined on a case-by-case basis. The MRHD in clinical trials with healthy subjects should not be equivalent to the maximum tolerated dose. However, in some patient populations, such as advanced cancer patients, the MRHD may actually indeed approach the MTD. So next we will discuss how MNS can influence non-clinical bioassay study designs. Guidelines for selecting an appropriate high dose for small molecule drug products is described by ICH M3R2. And there are 5 acceptable bases for high dose selection for general toxicity bioassays. They include the MTD, maximum tolerated dose, the maximum feasible dose, MFD, saturation of exposure, which includes saturation of absorption, the limit dose, which is dependent on the magnitude of the clinical dose, and exposure margin, where a 50-fold exposure margin is generally considered to be sufficient. When using exposure margin as a basis to select the high dose for non-clinical studies, this can be limiting later in development if the actual clinical PAD occurs at higher doses than originally predicted. So, which this could then necessitate exploration of higher clinical doses to achieve efficacy. And there have been cases where sponsors have had to repeat the non-clinical toxicology studies in order to be allowed to examine higher clinical doses and continue drug development, which of course can be very costly. Guidelines for selecting an appropriate high dose for biotechnology-derived drug products is described by ICH S6R1 and can also be based on the 5 toxicology, feasibility, and exposure bases for small molecules that we just talked about for ICH M3R2. However, PD endpoints, including the dose that provides the maximum PD effect or the dose that provides a 10-fold exposure margin, may also be an appropriate basis for setting the high dose in a non-clinical bioassay for a biotechnology-derived drug product, with, of course, the higher dose being the one that would be preferred. The PD endpoint basis for setting the non-clinical high dose can be justified when there are significant differences in PK/PD and biology between animals and humans, and when there are dose-limiting effects due to exaggerated pharmacology. In these cases, supportive use of PK/PD data, such as in vitro target binding affinity data and in vivo pharmacological data in which MNS can play a role in analysis and description, is also encouraged, particularly when in vivo and ex vivo PD endpoints are not available, but there is little or no activity in animal species, or when there are significant differences in PK/PD and biology between humans and non-clinical species due to different mechanisms of action. Apologies for the dog. I will now share a few examples of how dose exposure response curves can inform dosing regimen considerations in early phase clinical trials. This is an example of a linear dose exposure effect and toxicity relationship of an anti-diabetic drug with common hypoglycemia-related toxicities expected for healthy subjects on a normal glycemic background. The yellow box indicates pharmacodynamic activity, such as blood glucose lowering in this example, above the pharmacologically active dose, PAD. The red line indicates non-clinical doses associated with a high degree of blood glucose lowering, resulting in necrosis and mortality. The orange line indicates doses associated with neuropathy that, although reversible, are not monitorable. The orange shaded region indicates doses and magnitude of pharmacodynamic activity associated with non-clinical findings considered to be adverse. The green line represents the NOAEL for this drug. And based on the FDA 2005 guidance for estimating MRHD for first-in-human trials, the MRHD would be set at a dose 1/10 the NOAEL and fall well below the PAD for this scenario. Because there is a consistent linear increase in pharmacodynamic activity with increasing dose, and a consistent rate of dose escalation would be reasonable for phase 1 clinical trials. In this example, given that the dose-limiting toxicities are due to exaggerated pharmacology in healthy subjects, which includes the healthy animals that have a higher risk for drug-related effects, and given that subjects with hyperglycemia, such as the indicated patient population, would be expected to be less sensitive to the pharmacodynamic activity of the drug and have lower potential risk, there is sufficient justification to apply a less than 10x multiple safety factor to the NOAEL for hyperglycemic subjects. In fact, human equivalent doses approaching the NOAEL in diabetic patients, the indicated population, may be considered acceptable, especially given that the pharmacodynamic activity, i.e. blood glucose levels, can be easily monitored. This is an example of an exponential dose exposure effect and toxicity relationship of an anti-diabetic drug in the glucokinase activator, GKA drug class, which regulates glucose homeostasis within cells. As before, the yellow shaded region indicates pharmacodynamic activity above the PAD. The red line indicates the threshold for brain necrosis and mortality. The orange line indicates the threshold for neuropathy, and the orange shading indicates the threshold for adverse toxicity. Based on the NOAEL shown in green, the MRHD again falls well below the PAD for this scenario. Given the slow rise in dose exposure effect at low doses, larger increments with an initial rapid rate of dose escalation would be reasonable. However, given the steepness of the exponential increase in activity at higher doses, it is judicious to cautiously approach the NOAEL with small increments at a decreased rate of dose escalation. In other words, the steeper the curve, the slower the rate of dose escalation. This is an example of a logarithmic dose exposure effect and toxicity relationship of an anti-diabetic drug in the sodium glucose cotransporter SGLT inhibitor drug class, which prevents renal glucose reabsorption back into the bloodstream. In this example, you can see that the pharmacodynamic activity approaches an Emax as the curve flattens out at higher doses. While renal tubule dilatation is observed at lower doses, it is likely secondary to the pharmacodynamic activity of the drug in the kidney and is not actually considered to be adverse. However, at higher doses, adverse findings in the non-clinical studies include renal toxicity and kidney failure. In this example, the calculated MRHD would be based on 1/10 of the NOAEL, is actually higher. So let me restate that. The calculated MRHD, which is based on 1/10 the NOAEL, is actually higher than the PAD. Thus, this would be an example where it would be more appropriate to determine the MRHD, maximum recommended starting dose, based on the anticipated PAD, which of course comes from the MNS. Since the curve is anticipated to be steep at low doses, and since a primary goal of phase 1 studies is to accurately describe the pharmacodynamic activity of a drug, an initial slow rate of dose escalation would be appropriate. Otherwise, one would completely miss the characterization of the drug's pharmacodynamic activity. But it would be appropriate to be followed by an increased rate of dose escalation as Emax is approached. Again, the steeper the curve, the slower the rate of dose escalation. So I'll now take a few minutes to mention reformulations, where there are several areas where MNS can play an important role in drug product formulations. It is common for drug products to undergo reformulations throughout drug development during the lifetime of an investigational new drug, IND application, with ongoing clinical trials prior to the market application as an NDA or BLA. As formulation changes occur, MNS modeling can be useful in evaluating and demonstrating bioavailability and bioequivalence. The 2015 FDA reformulation guidance states that an adequate evaluation of the pharmacokinetics and absorption, distribution, metabolism, and elimination, PK/ADME, of the drug substance is recommended for new formulations. However, it should be noted that additional non-clinical bridging studies may be warranted. Nevertheless, MNS simulations demonstrating bioavailability and bioequivalence can be useful in justifying the sponsor's position. For reformulation of approved products for extended release formulations, an in vitro in vivo correlation, IV/IVC, may be useful as a surrogate for in vivo bioequivalence when it is necessary to document bioequivalence. And this was described by the FDA extended release guidance. And this could be a significant cost savings for drug development. Switching to drug accumulation potential, since we still have a few minutes, I'll attempt to highlight some aspects of this challenging area where MNS could potentially play a role and provide some useful insights. So there are several instances when distribution and accumulation may need special consideration, which can present regulatory challenges. This includes when there is significant distribution to tissues with relatively slow clearance rates. And this can then lead to accumulation. When there are increases in pharmacodynamic related effects with repeat dosing that could be secondary to significant distribution and or accumulation in specific target tissues, and when there are concerns that the preferential tissue distribution or accumulation could be related to the tissue toxicity. And this becomes particularly important if it's related to adverse toxicities that affect the NOAEL determination and the overall maximum recommended human dose allowed. And a good example of this is the distribution of oligos to target organs with toxicity, and I'll talk a little bit more about that example. Redistribution of drug released from tissues can also affect PK/TK parameters in other compartments, and we see this with the slow release of lipophilic drugs from fat compartments, as well as the slow redistribution of oligos from target tissues, and I'll talk a little bit about that. So as an example of drug accumulation potential, I'd like to share with you some observations for the systemic phosphorothioate antisense oligonucleotide drug class that I'll refer to as PSASO. This drug class usually has broad tissue bioavailability and similar patterns of distribution throughout the body, and common plasma PK characteristics where the Tmax is actually very short, and the drug is rapidly taken up by the tissues, and however, there is a very slow tissue clearance, and as you can see in this example, that there is actually much more drug present in the tissues after 24 hours and practically nothing in the plasma. So the drug is rapidly removed from plasma due to rapid tissue uptake and very slow tissue clearance. Continuing with this example, it is readily apparent that there is biphasic plasma PK with a rapid rise and fall indicative of the distribution phase. And this is likely due to the endocytosis into the tissues. Across species, this can range from minutes to hours, but in general, by 24 hours, it is pretty much gone from the plasma. In the elimination phase, which can be described by a very prolonged return to baseline, this phase is actually likely to be related to the redistribution phase, wherein plasma concentrations come into equilibrium with tissue concentrations. That is likely driven by the slow exotosis from the tissues. Across species, this can range anywhere from days to months. Overall, I also wanted to mention that if you look closely and considering the long time scale of this PK graph, there may even be a third phase, but based on that time association. But overall, a standard 3-compartment model, which is used for most drugs, may not be sufficient to model these types of drugs. Taking a closer look into how this might look for a PBPK model, it's readily evident that there are actually many compartments to consider for this drug class, and it quickly becomes very complicated. So if Kn in blue represents the rate constants for the drug being taken up into the tissue, then the purple K values could represent the rate constants for the tissue clearance. You can see how there's a significant imbalance in with PSASO repeat dosing, plasma C trough steady state values aren't achieved until at least 3 months. So there are many variables and unknowns, many compartments, and many different rate constants, and as well as a high degree of interspecies variability. So the question becomes, is it then impossible to predict for these types of drug products with this significant drug accumulation in tissues? And this then leads to the next question for regulators, which is AUC exposure for dose margin determination still the best? In general, AUC exposure is our gold standard. For the systemic PSASO drug class, steady state is identified by looking at plasma C trough levels. This is because the acute increases in plasma exposure levels with dosing are less informative and do not accurately describe the pharmacodynamic responses. A constant consistent challenge is that the acute plasma exposures do not necessarily reflect the target tissue exposure and cannot be assumed to be comparable between species. In fact, steady state levels are often not achieved until phase 3 clinical trials because it can take 3 to 6 months to achieve the steady state plasma C trough, at which point there is no longer further impact from drug redistribution. Consistent with this is that it also takes 3 to 6 months to achieve steady state pharmacodynamic activity and 3 to 6 months to complete plasma elimination, as well as 3 to 6 months for pharmacodynamics activity to return to baseline levels. So this also becomes very challenging in determining the best clinical doses prior to time to submit a new drug application. The prolonged time to achieve steady state is likely driven by the significant distribution and accumulation effects. And overall, what this comes down to is that the value of PS-ASO plasma AUC exposures for dose margin comparisons of toxicities and target tissues is very limited. Since the gold standard for regulators is to determine dose margins and safety, this brings about the question, are AUC exposures to dose margin determination still the most appropriate choice? And if not, what might be the appropriate alternative for the safety dose margin determinations? The FDA 2005 guidance briefly mentions ASO oligos, where it talks about complement in the blood compartment and states that for some antisense drugs, the Cmax correlates across non-clinical species with mg per kg dose, and in such instances, mg per kg scaling would be justified. In other words, Cmax and body weight bases are appropriate for the blood component. But what about drug-related toxicities in the tissues, which are usually the dose-limiting adverse toxicity for these drug products? So I'm going to share with you some observations from the Volanesorsen BSA basis determination as an example, and this can be found in the 2018 advisory committee document. So this drug has the several contributing factors similar to what I've just discussed, with the short plasma exposures despite prolonged pharmacodynamic activity, increases in C trough values with repeat dosing, but where steady state wasn't achieved until 3 to 6 months later. Also had the prolonged tissue half-life, particularly in target organs of toxicity, such as the kidney and liver, and where the drug accumulation was considered to be a likely contributor to these adverse toxicities. And of course, there were several complexities in the oligo tissue pharmacokinetics and compartmentalization, like I just discussed. Overall, this was determined to decrease the validity of comparability of the AUC exposures to the species, resulting in an unclear exposure relationship to the animals. And the regulatory scientists determined that the BS body surface area correction was actually the most appropriate basis for organ toxicity for this drug. And this led to quite the debate, as the sponsors had a much larger margin of safety based on AUC exposures and little to no margin of safety based on BSA. In the Mikkel-Mersen Advisory Committee meeting in 2012, Dr. Wang did a really nice interspecies comparison, taking organ toxicokinetic data, and was able to demonstrate that in those organs, a BSA correction was indeed the most appropriate. And that was also another systemic PS-ASO, and that was part of the reason why a similar approach was taken for Volanesorsen. And it's also important to keep in mind that both the blood compartment and the organ systems can contribute to the mechanism of action. So this brings us back to this question. Is the BSA basis applicable to other drug products with significant PK-TK contributions due to distribution and/or accumulation? And this remains a matter of debate. So in summary, we've talked about how modeling and simulation can be useful in predicting, and is very useful in predicting clinical pharmacology, MABEL, PAD, and ATD prior to first-in-human trials. For clinical doses, the maximum recommended starting dose and maximum recommended human dose are determined based on empirical non-clinical data, but not on PK or PBK modeling. However, the MNS-derived pharmacology endpoints, MABEL and PED, can be appropriate for MRHD determinations for some drug products. And also, the MRHD, it's important to remember that the maximum recommended human dose is based on empirical non-clinical findings, not the MNS, and can be limited by non-clinical bioassay high-dose selections. However, again, it's important to note that the MNS can play a role in providing supplementary information to support high-dose selection for non-clinical bioassays, particularly for biotechnology-derived products. Also, we talked about how dose exposure effect curves and toxicity can inform dose escalation decisions for early phase clinical trials, and how MNS can play an important role in reformulations and support bioequivalence submissions. We also talked about how MNS information related to drug accumulation potential could be useful to regulators for some products. And this is an area of where there remain many knowledge gaps. That's all that I had today, and I apologize for going over and all of my connection issues, but thank you. I'm happy to take any questions.